Okay, so let's get started. It's 9 o'clock. Thanks everyone to join us during the weekend, and in particular, the big thanks to our today's speaker, Dr. Han Shih, to actually save the day today to replace our original speaker. Uh, so really appreciate it. So so it's really my great pleasure to welcome our today's speaker, Dr. Han Yixi, and also my colleague from National Eye Institute, National Institute for Neurological Disorders and Stroke, from NIH. So Hai actually got her undergrad study in microbiology from National Taiwan University, and then she pursued her PhD training in Department of Immunology from Duke University. So during her PhD training, she mainly interested in studying the role of three-dimensional chromatin looping and the key looping factor CTCF in gene regulation during thymocytes development. So from 2013, she actually decided to move to NIH and join John Shih's lab in NIAMS, where she starts to continue sort of a continuation of her PhD study and study the lymphocytes regulatome as a postdoc fellow. She actually really did a very fruitful postdoc work and then published a series good papers. And then from 2019, she started her independent research program as a joint appointment by NEI and NDS as a tenure track investigator. So Hai's lab is really interested in understanding the mechanism which how they precisely regulate gene expression in lymphoid cells through multidisciplinary genomic approaches. Her research focused on understanding the regulation of cytokine loci in immune regulosomes in health and neurodegenerative diseases in both eye and the central nervous system, with a with a goal of developing more specific and effective therapies. Previous work, Hanye's lab have identified different lineage-specific regulosomes in tissues resident innate and adaptive lymphocytes in both homeostatic and then activated states. And then currently, her lab focuses on two key biological questions, which including how do distinct stimuli regulate cytokines in a context-specific manner. And then the second question is how cytokines contribute to the progression of neuron degeneration, such as Alzheimer's disease and age-related muscular degeneration. By analyzing lymphocytes regulosomes with distinct genetic background in human and engineered mice, Heinz lab aimed to further the understanding of molecular machinery that contribute to aging and neurodegenerative diseases. So as I mentioned, Hai really actually made a lot of groundbreaking discoveries, and then based on that, she received numerous awards and honors, which including NIH, NIID Career Transition Award, K22, and then an NEI Direct Scientific Innovation Award, multi-institutional synergistic idea award from this Department of Defense. And this year she got NEI Directed Awards, NEI Data Management and Sharing Champions. So thank you so much, Hai, to join us today on our platform. Really looking forward to a talk. Please go ahead.
Thank you, Chan. Thank you for your invitation and such a very kind introduction. Actually, it's really my pleasure to be here because I've heard Immune Zoom for a long time, and finally I have the chance to present here and to interact with people all over the world. So, as Chan mentioned, we are interested in the lymphocyte regulomes, trying to understand how the chromatin landscape facilitates the regulation of immune cells, both in homeostasis and the diseases. And let's see, next slide. So this is a art created by ChatGPT by Dell, which indicate the fundamental concept about the immune cells that require a very complex regulation that connect the environmental changes to immune functionality. So to this point, I think this is a very common concept about why we know the heterogeneity of lymphocytes. It's also simplified and missing other key components. But the point I want to highlight here is the lymphocytes, depending on their development and their functionality, they can be generalizedly categorized into adaptive and innate component. So adaptive lymphocytes, as you know, has to recognize the antigens from antigen-presenting cells to be further differentiate, while the innate cells have developed their functionality through their developmental stages. And the other way to look at these lymphocytes is to base on their functionality. So the type 1 immune cells, including cytotoxic T cells, CD8 T cells, Th1, K cells, ILC1, those are key interferon gamma producers. While the type 2 immune cells, including Th2 and ILC2, produce type 2 cytokines. The type 3 cytokines, IL-17 and IL-22, are produced by different subsets of adaptive and innate lymphoid cells. So not only these cells can produce similar cytokines, the regulation of these immune cells are also shared between adaptive and innate lymphoid cells, particularly if you think about the transfer factors, the master regulators of these lymphocytes. So one very interesting question I have been always intrigued by is how individual cell that contain 6 billion nucleotides shared across the cells can behave so differently and carry so many different type of functions. So how that lineage identity and the functionality are determined by the DNA chromatin status. So this is also another analogy that I like we proposed a while ago. So the individual aspen trees here look very different. So they are like innate adaptive lymphocytes, but the fundamental, their chromatin, the DNA sequence are identical, which is actually the concept of aspen. They share the same root, even though they look like individual trees. So the way we're trying to understand how the individual cell are shaped differently is to understand their root, the regulomes that are determined by the combination of signals and the transfer factors. So why regulomes are important? As many of you may have known that among our 6 billion, so 3 billion doubled by duplicate, the 2% coding regions is very critical for the functional proteins. However, around the 98% DNA sequence are non-coding regions. These non-coding regions are not there being useless, but actually they are very important to switch on or off or fine-tune the transcriptional labels in a context and a stage-specific manner. So when I talk about non-coding regions, those are like functional DNA elements such as enhancers. They can target specific genes independent of the sequence in the genome. And it has been well characterized over 2.8 million regulatory elements in the database, which is important to control 20,000 genes that are coding regions. And it has been shown that over 90% of disease-associated SNP are located in non-coding regions, suggesting the non-coding regions play a very critical role in gene regulation. Especially, it's important during immunoresponse, as we need very precise and timely immune response to control the pathogens without inducing the undesired autoimmunity. So luckily, we have a lot of tools, methods has been developed to tackle this question. So single-cell RNA-seq and the RNA-seq allow us to understand the output of this regulation, while the ATAC-seq, which replace completely the old DNase method to identify potential regulatory elements that are often open or more accessible for transcription factor binding. There are also methods such as ChIP-seq, cut and run, the cut and tag. Those are incredible methods allowed us to tackle the question, what's the status of the chromatin that allow them to regulate the target genes. And another layer of the regulation I will be touching on today, which is through the looping formation that allow the enhancers regulatory elements to physically modulate the gene expression on their target genes. So here is how we think about a cell. So in the cell, the nucleus, it's like a supercomputer. They can compute a lot of signals from the environment. It could be growth factors, cytokines, or even neuropeptides and the metabolites. These signals can carry the messages by the message from these signals are carried by transcription factors into the nucleus, and depending on the local environment that's and the chromatin status, they will produce different kind of output. The output, majority of the outputs are the cytokines, which can determine the barrier integrity and communicate with other immune cells for the maximal activity of the immune response. And recently, there's also more and more evidence showing there are also neuroimmune crosstalk. So these events are very important for lineage commitment and also cell plasticity. They are also very critical to induce cell activation and also result in the senescence of the cell status. This is also very important in terms of determining cell standness and also the exhaustion level. So by saying that, how do we tackle these questions and how do we conclude from this very complex scenario, which is actually a very simple formula. You have at the time point, what's the status of your chromatin and what are the transcription factors available. And then the combination of these events will also be determined by surrounding factors that result in the final output. So we are trying to understand this chromatin status, and we initiate with a very simple method, which is ATAC-seq. So ATAC-seq allows us to profile the overall regulatory elements in the cells. Now we can do it even at single cell level. So in the beginning, we have profiled over 20 different immune lineages. We found, as you can see, the clustering here. Even though ILCs and the T cells, they share the functionality, they are actually clustered separately. This is a paper from our first cell paper in 2016. However, they are distant from other immune cells, suggesting indeed, even though they are ILCs and T cells, they have some commonness in terms of chromatin landscape. So we next asked, what's one thing I didn't mention is the ILC is very unique. It's because their chromatin landscape are shaped or rewired as soon as they commit to specific lineage. So the ILC progenitor already has primed chromatin that's been carried during their differentiation and allow them to rapidly respond to the environment. So how does the rapid immune response occur in the innate lymphoid cells in a very short timing? This is another study we focus on trying to understand the chromatin dynamics in the NK cells using IL-22, IL-12 and IL-2 post-stimulation, which is now a very strong signals that induce interferon gamma production through STAT4 and STAT5. Without going to the details, since this has been published, we found there's a lot of crosstalk between lineage-determined transfer factors, the T-beta transfer factors, and the signal-dependent transfer factors, the STATs. So what happened is, upon the very short time stimulation within hours, we can see both poise enhancers and the de novo enhancers are required for short-time gene activation. So what are the poise enhancers? The poise enhancers are the enhancers that's already been modified during NK cell development. However, upon stimulation, the STATs will come into this region to allow final terminal activation of these enhancers, which can be detected by P300 transcription factor binding and also the marker, the K27 acetylation. In contrast, the de novo enhancers are those that inaccessible chromatin during development. However, upon stimulation, the STATs will target these regions to induce the chromatin opening. And this process also allows the recruitment of lineage-determined transfer factors such as T-beta to these new enhancers. And then based on the P300 modification, they will be rapidly activated in a short time, within hours. So now we know the already primed innate lymphoid cells has to undergo rapid chromatin remodeling through primed poised enhancers and also the de novo enhancers. We asked, is this permanent or not? Which has been answered by Zhou Sang's group. They use the MCMV infection model to show indeed there's a rapid opening of chromatin landscape in NK cells upon infection. However, this is a temporary shape. Majority of them will be closed down and remain some memory on the chromatin, that's so-called NK memory. And next, the comparison, we want to understand how the T cells differentiate. And also, we found something interesting is even though the developmental process is very different between innate and adaptive lymphocytes, actually, upon infection, there's a synergy of a convergence of the chromatin landscape. So you can see they share a lot of chromatin landscape after T cell differentiation. So, okay, so that's a little bit just a background, like how we see the innate and adaptive lymphocytes. They develop their functionality related chromatin landscape over time. So during this time, we are interested in the genome-wide study, but at the same time, we found something very interesting, which is kind of known, but maybe less understood. So for example, the genes that function in the same immunity they are often nearby each other to be co-regulated, such as IL-4 and IL-5 and IL-13. They reside within 150 kb region that can be easily co-regulated. Also, IL-17A and IL-17F are co-localized in the genome, which allow them to be co-regulated. What's very interesting is interferon gamma gene, which is a highly regulated gene in type 1 immunity, is nearby IL-22 in the genome. So this is the one, the locus cover the IL-22 and interferon gamma, which starts from NDN1, which is a non-immune-related gene, and end at interferon gamma X1, the long nucleotide RNA, that's known important for terminal Th1 differentiation. So interestingly, when we look at the chromatin accessibility by TAC-seq, we found although interferon gamma relies on a cluster of enhancers surrounding it, these enhancers are only accessible in the cells that produce interferon gamma, such as NK cell, ILC1, and Th1. And in contrast, IL-22, although nearby interferon gamma, the chromatin landscape is only decorated in the type 3 lymphocytes, such as LTI and ILC3. So this landscape has fit the category of the enhancer landscape called the super enhancer, which is defined by Rick Young's group. So our question is, since these locus are so differentially regulated in different lineages, whether the three-dimensional chromatin loop that's important for this regulation. Another interesting thing we found is the synteny of this locus is actually pretty conserved across different species, as you can see here. And the MDN1 gene is actually defined as a duplication-resistant gene, which support the idea the chromatin looping might be very important for regulation of this locus. So we really want to tackle the question whether the higher order chromatin architecture contribute to the Th cell differentiation. So just a little bit more background about the chromatin architecture. As I mentioned, individual nucleus contains 6 billion nucleotides. However, they are not like this plate of spaghetti randomly distributed in the plate. But instead, they form these modules of the plastas in the plate, which so-called topologically associating domains. These domains are very critical because they define the numbering of the regulatory elements. So the enhancers can precisely target their genes without getting confused. And this loop formation has been proposed to be regulated by the cohesion complex that slide on the chromatin and pull out, extrude the DNA to form the loop, but stop at 2 CTCF binding sites that have convergent orientations. So the CTCF sites are very important as anchors of TAT, which is a very important idea in this kind of research. And again, luckily, because of your Decker's work, now we have perfect method to detect the DNA chromatin that's associated with each other to form the loop independent of their linear distance. So the Hi-C and the related method allow us to detect 2 regions of the chromatin that has higher frequency of interaction, which can be shown by this triangle demonstration. So to understand whether the loop formation is important for differential gene regulation across interferon gamma locus, we perform the Hi-C on the naive CD4 T cells and the Th1 cells. This is performed by my former scientist, Chenhong Liu, with 2 postback, Sadie and Will. So what we saw is, compared to naive T cells, as expected, there's a lot of interactions across interferon gamma region to form the interferon gamma tag. What surprised us is there's also a new tag formed upon Th1 differentiation. This IL-22 tag starts from the CDCF site within the MDN1 to a CDCF site that's downstream of 70 kb downstream of interferon gamma. This is the strong CDCF site that seems to form the boundary to segregate the IL-22 and the interferon gamma tag. So to prove that's the right idea, we then one thing to mention is based on the orientation, we also believe the IL-22 tag is formed by CDCF that has convergent orientation. We then further generate a mouse that have this CDCF binding site deletion, because sometimes if there's a mistargeting, the result might be a little bit difficult to interpret. So we only target 17 base pair on the core of CDCF binding site to understand this phenomenon. And before I show you Th1 data, I want to also remind you the Th1 differentiation requires stepwise signals. So first, the naive CD4 T cells has to encounter dendritic cells through TCR to get the TCR signal and the co-stimulation. And this allow the low-level IL-2 and interferon gamma production that's trigger the lineage determined transcript factor TB expression. And this initiates the Th1 differentiation because it induces more interferon gamma producing, and at the same time, these cells can sense the signals from the myeloid cells, such as using the IL-12 that stabilize the lineage identity. So to ask whether the CDCF binding site is important for this process, we perform Th0 condition, which mimic the signal 1 and 2 stimulation, and the typical Th1 condition that's at the IL-12, the key driver of Th1 differentiation. We also use Th2 condition as a control. As you can see, the Th1 cells show the deficiency in interferon gamma production in the knockout mice. However, even though the interferon gamma level is undetectable in Th0, we do see a deficiency of TB expression. And we know by blocking interferon gamma, this process is dependent of low-level interferon gamma to proceed. And on the right is the quantification of the result. So this data suggesting this CDCF binding site is really critical for initial Th1 differentiation. To understand whether this is a very targeted event or the Juno-1 event, we then perform bulk RNA-seq on the Th1 cells differentiate for 3 days and the Th1 cells differentiate for 7 days. We found that indeed interferon gamma is the key gene that's downregulated in the knockout Th1 cells. Interestingly, we also see slightly downregulation of interferon gamma AS1, the long non-coding RNA that's important for terminal Th1 differentiation, suggesting there's a defect in Th1 differentiation in this CDCF knockout mice. Another interesting thing is on day 3, we also see slightly induction of type 3 signature genes, such as CCR6 and IL-22. And using the more experiments that I'm not going to show you here, we know that the IL-22 defect is intrinsic, meaning it's through the locus defect that failed to regulate IL-22 precisely in the Th1 cells in the knockout. So we conclude that this CBS -70 CD3 binding site is essential for initial Th1 differentiation. Another interesting thing we found is whatever we stimulate NK cells, which also important interferon gamma producer, there's no difference between the wild type and knockout, suggesting the innate adaptive lymphocytes, they can be regulated differentially, and only the innate cell, adaptive cells require this loop formation, while the CD3 binding site is dispensable for interferon gamma production in NK cells. So since now we see a phenotype from in vitro assays, we next ask whether this is important in vivo as well. To answer that question, we perform single-cell RNA-seq data on the mice that infected by Toxoplasma gondii, which is a very strong type 1 immune response inducer. We analyze the cells isolate on day 3 and day 7. And here is the result. You can see, indeed, in the T cells, so S15 specific T cells is antigen specific T cells, we see a dramatic reduction of interferon gamma, as well as their target genes, IPTG and ITIF3. However, again, in the NK cells, there's no reduction of interferon gamma production. And then this is, again, support what we've found in vitro, that the CD3 binding site is important for T cell, but not NK cell interferon gamma production. Another interesting thing is we also found the Th17 signature genes are only upregulated in the knockout mice, but not in the wild type mice. And this specifically occur at early stage on day 3, but not on day 7, suggesting the deficiency of Th1 commitment is only occur in the early stage. What's more important is whether this immunodysregulation contribute to the disease control. And then the answer is yes. So when we infected the wild type and the CBS -70 knockout mice, we found there are more infected cells in the knockout, and the weight lose faster in the knockout. Potentially, you can see it's at earlier stage, but then comparable later on. And also we see less survival rate from the knockout mice, suggesting even a very subtle dysregulation of Th1, Th17 differentiation. We can see a phenotype from the Toxoplasma gondii infection. So what we are interested is what's the molecular mechanism behind these phenotypes. So we again performed a high-C assay to detect the chromatin interactions. We found indeed there's a huge reduction of the interaction that's shown previously from the IL-22 TAT. More interestingly, there's an increase of interaction of MDM1 to the region that's showed as a CD3 binding site. So this -30 CD binding site has a weaker CD3 binding, but then increased, probably replaced the -70 CD3 binding sites after it's deleted. And this in contrast, we see there's no difference in terms of the interactions on the right side, the SE2 part. However, we do see slightly increase of the interactions between the SE1 the super enhancer 1 with IL-22. And this might explain why we also see partial or low level of induction of IL-22 at early stage. So why there's a difference between the Th1 NK cells for this CTCF binding site dependency? So here is how we understand about Th1 differentiation. So from day 1 to day 7, during the initial Th1 differentiation, the enhancer landscape started to be modified surrounding SE1, the left side of super enhancer, and gradually goes to the right SE2 at the later stage. So there's a progress of the enhancer modification, which somehow rely on the CTCF binding site next to it. In contrast, the NK cells has established their enhancer landscape early on during development, as I mentioned earlier. And when getting activation, they utilize multiple enhancers rather than just depend on the left end of the enhancers. So this might be the reason why the NK cells doesn't require the CTCF site that's marked the left end of the super enhancers. And indeed, when we look at the loop formation in the NK cells, we do not see the IL-22 tag in the either wild type or in the knockout. So this is our model. We think in the wild type Th1 cell differentiation, there's two TAT formed to allow differentially regulation of this IL-22 and the interferon gamma. While when we delete this -70 CTCF binding site, it introduces a new TAT that separates the super enhancers and partially interact with IL-22 to induce dysregulated cytokine production, while in the NK cells, because there's no IL-22 TAT, there's no significant difference after activation. This is another way to look at the three-dimensional loop formation. In the wild-type Th1 cells, the DNA topology allow the interaction of SE1 and the SE2 to their target gene interferon gamma. However, when we delete the -70 kb region, the cohesion will further find the next CTCF binding site, -30, which separate the SE1 and help the IL-22 expression, but reduce the interferon gamma production. So to summarize the first part I've talked about, we have shown you that the lineage identity and the functionality can be defined by the regulomes. And these regulomes in the innate cells and in the adaptive lymphocytes are developed in a very different way. So in innate cells, they are primed, and that allow them to rapidly respond to the immune response. And this is less dependent, especially for interferon gamma, less dependent on the CTCF binding site next to it. While in adaptive lymphocytes, such as Th1 cells, it requires stepwise dramatic chromatin remodeling, including loop formation. And this make the 3D structure more important for Th1 differentiation, which is different from innate lymphoid cells. So for the last couple minutes, I want to switch gear a little bit. So as I mentioned, we are very interested how the chromatin sense the signals from the environment and determine the ultimate output. And then, as you know, in the individual tissues, the environment can be very different. And therefore, the same immune cells can function or behave very differently in different tissues. Recently, we are more interested in the CNS because CNS, the brains and the eyes, have been considered as immunoprivileged organs. And then more recently, we have found, people have found, and there are immune cells in the CNS under non-healthy situation. So we are very interested in the crosstalk between these immune cells and the neurons. So to initiate this understanding, we first utilize a mouse model that developed Alzheimer's disease. So Alzheimer's disease, as you know, is the leading cause of dementia. The major pathology including amyloid beta plaque formation and the tau protein neurofibrillary tangle formation in the neurons. This will lead to synapse loss and neural death, and unfortunately will cause cognitive impairment. Sadly, we have very limited options for Alzheimer's disease treatment. The most recent promising treatments are focusing on using antibodies that against amyloid beta plaques to clear these plaques. But this treatment is only show the effective initial stage of the disease. So we are interested in downstream the pathways. Maybe this is not sufficient for disease control. So in the brain, especially in the Alzheimer's disease brains, there are a lot of pathology that's surrounded by different cell types, such as neurons, astrocytes, oligodendrocytes. And among these cells, the microglia is the key immune cells that's identified in the CNS. And previously, Ido Hami has showed the disease-associated microglia that's very critical in aging and in neurodegenerative disease. So we are interested in lymphocytes or other non-microglial immune cells. So to ask whether lymphocytes or other non-microglial immune cells contribute to neurodegenerative disease, we utilize this animal model called the 5XFAD, which carry 5 different mutations on 2 important Alzheimer-associated genes, APP, which is the amyloid precursor protein, and the PSEN1, which is the enzyme that digests the peptide. This mouse can develop the phenotype that's similar to human Alzheimer's disease, including forming the plaques and also have the synaptic loss and the neural death. So when we start this study, it really remind me the art of Vincent van Gogh. So this is the Starry Night, which Vincent draw the stars among his crazy brain. When we first look at the plaques in the amyloid beta transgenic mice, we see these plaques formed in the brains that really like the stars in the Starry Night, except this is very sad as the initial factors for Alzheimer's disease. Interestingly, we did find the immune cells that infiltrate to the Alzheimer's disease brain. And this is very abnormal because you don't see any immune cells other than microglia when the mice or when the human are healthy. So to unbiasedly understand the immune cells in an Alzheimer's disease mouse model, we performed a single-cell RNA-seq on the cells isolate from wild-type and 5XFAD brains at different stages. We focus on CD45 high cells that easily help us to segregate microglia from other non-microglia cells. Here is the result to show the proportion of immune cells, key immune populations in the single-cell RNA-seq data. We found along aging, there's a dramatic increase of T cells at late stage, around 12 months old wild-type mice. However, when we compare the wild-type with the 5FD mice, we found that this phenomena occur earlier, around 6 months, suggesting 5FD has a accelerated aging phenotype. We then call this disease-associated T cells based on ADAM's nomenclature strategy. So what are these stats? Comparing the wild type and the 5AD, we found the majority of the stats are the CD8 positive T cells. These CD8 positive T cells can be even categorized into 3 different subpopulations. The third population remain consistent over time, while the subset 1 to 2, they are induced at mid-stage, and the subset 1 kept to be high at the late stage. And we are not the only group found this CD8 T cells in Alzheimer's brains. Both in mouse and human, people have shown the T cell infiltration that's allow them to communicate with the microglia for the pathology of neurodegeneration. But we really want to dig into how these CD8 T cells are regulated and how they contribute to disease. So to understand that, we performed a deeper single-cell analysis focusing on CD8 T cells in the brains. We found there are multiple clusters, including cytotoxic CD8, naive CD8, and also an interesting group which highly express interferon stimulated genes. And we found during aging in the wild type, we lose cytotoxic and the naive CD8 T cells, but the cells stay in the intermediate stage, which difficult to classify, but that's why we call them class 1 and 2. However, compared to wild type, the 5FAD mice, they further differentiate into this ISG positive population, which is dramatically increased as compared to the wild type. So we believe that has a very strong interferon stimulated phenotype, which suggesting there's a very strong inflammation in the brain. We also perform trajectory analysis and show there's 2 major cell fate along aging and along the disease progression. So the naive T cells can differentiate into exhausted stage as well as ISG positive stage. And without showing here, we do see the ISG positive cells are mainly from the 5FAD mice. So next question we have is whether these cells really are in the brains and how do they interact with other cells in the brains. So to answer that question, we perform multiplex immunohistochemistry. We utilize different markers to target immune cells, blood cells, and also glial and the neurons, as well as different pathology, including amyloid beta and phosphorylated tau. As you can see here, compared to the wild type, the 5FAD mice showed a lot of beautiful stars in the brain, which is the pathology, and that triggered the inflammation. When we zoom in, we can see the CD8 T cells infiltrate into the hippocampus, and we can sometimes see 2 cells are nearby each other, suggesting there might be proliferation going on. So here's a bird view of how cells are distributed in the brain. So compared to the CD4 T cells in both wild type and the 5AD, they can only be found surrounding the ventricle or in the meninges. The CD8 T cells compared to wild type, they are incredibly increased in the parenchyma of the brains in the 5AD mice. So on the right is the statistics. And if we look closely to this CD8 T cells, we do see they are not only can interact with microglia, the major immune cells in the brain. We also see association between CD8 T cells with plaques, with astrocytes, with neurons. So there are many different events going on when this CD8 cells comes to the brains. So there's still a lot of unknown interactions that's waiting for us to understand. One important thing is we don't see a relationship between the CD8 T cells often with the blood vessel markers, suggesting these CD8 T cells indeed left the blood vessel and infiltrate brain parenchyma to become tissue-resident immune cells. So then we want to ask whether these stats are important for amyloid beta plaque formation and whether it's important for cognitive impairment. So to answer that question, we perform IGC on the 5FAD mice and also the mice that cross to the B2N deficient background to prevent CD8 T cell development. We found that indeed there's a dramatic reduction of plaque formation in the 5AD B2N knockout mice. On the right is the quantification of plaque area in cortex, hippocampus, and other regions. We also performed the behavior assay, which we use Y-maze assay to determine whether there's changes in terms of mouse memory. We found compared to the wild type mice, those have more tendency to try new arms or in the mass to show higher percentage of alternation. The 5AD mice, as shown previously by other groups, they show lower percentage of alternation. Interestingly, when we perform the same analysis on the 5FAD B2 knockout mice, we do see they actually recapitulate the memory as the wild type. And this is the phenotype. There's a quantification we got for the Y-maze analysis. As you can see, the 5FAD B2 knockout mice has higher spontaneous alternation as compared to the 5AD only. So to summarize the last part of our research, we've using the single-cell transcriptome analysis to show disease-associated T cells in aging and the 5AD brains. And then this population features tissue-resident memory lymphocytes with interferon-stimulated gene signature. While if we try to remove CD8 T cells, we actually prevent the development of memory loss in these mice. I would like to thank everybody in the lab who helped to build different projects from different perspectives. Especially, I would like to thank Chen-Hong Liu, who is the lead for the CTCF interferon gamma regulation project, and then Alicia Fernando, who was the lead for the Alzheimer's disease immunoprofiling. And I would like to thank Janan, who is taking care of Alicia's project, trying to understand the regulomes in the CD8 T cells, and also performing spatial transcriptomes to identify, pinpoint the deaths in the brain spatially. So I also like to thank all the collaborators, and I would like to thank you for listening to my talk at this very unique time frame. I would like to take any questions.